Good afternoon, everybody. I'm Luciana Migliore from the University of Rome Tor Vergata, and I would like to speak about our studies about the microbiome of seagrass and how to go from the community structure to its function. So to start, we will start, as you can imagine, by speaking about the seagrass importance and way of monitoring. Then I will give you a short introduction to the concept of seagrass holobiont, then how to make the microbial community analysis, then I will show you four case studies regarding mainly Alophila stipulata, but also other native Mediterranean species. And after this, I will go through the conclusion. So, just to start, you all know why seagrass are so important, and they are very different species from the most ephemeral, like Alophila, to the climax species, persistent climax species like Posidonia, or intermediate like Chimodosia. And they are all really important because of the high capacity to stock carbon, the one that we call usually blue carbon, for CO2 sequestration, for sediment stabilization and coastal protection, and this is really true for Posidonia, but also because they are food resources for a lot of animals, as you can see in the pictures, and also shelter and nursery for juvenile fish and crustacean, even of economical importance, nutrient uptake, so water filtration, and all these ecosystem services in economic terms has been evaluated as 2.8 10 to 6 US dollars, so a huge amount of money for a really important service of these systems. Actually, although they are important, they are really treated. We have already lost about 30% of the historically known meadows because seagrass are really sensitive to environmental changes and to human-induced disturbance. And actually, global loss of seagrass, it's much higher than the loss of tropical forest. Obviously, to monitor the ecological status of seagrass is a very pivotal point in both research and management policies, and the choice and combination of measurable and sensitive indicators it's the only way to have a complete understanding of the effect of stressor and for a rapid and effective response. Since the methodological approach is very important, we decide to have an integrated approach for seagrass monitoring that starts with the environmental variables, obviously, like turbidity, sediment type, slope, nutrient pH, temperature, to pass to the biometrics measurement that are very common, like meadow density and cover, biomass, leaf morphometrics. But although they are very common, this kind of descriptor take some time to show their response. There is a lag from the stress to the response, And there is also a lag from the recovery to the response. So they are not so useful. They are very important, but are not useful for a rapid intervention. Much more interesting in this sense are the physiological descriptors like photosynthetic pigments and total phenols that always according to Roca et al., are early warning indicators. That is, they gave an early response to degradation, and actually they give also an early response to recovery. So they are very, very important and useful. Actually, to conclude this integrated approach, we included also the ecological interaction, and actually the one that I consider very important is the study of the epiphytic microbial communities that move from the Seagrass ecological state of measurement to the evaluation of the Seagrass holobiont. What is a Seagrass holobiont? Actually, all the stories start with the corals. There is a basic paper, two basic papers that you can see there of Rosenberg and Zilber-Rosenberg that started this evaluation by studying corals, by studying the combination of animal corals and their microbial associate. And they discover that this is a very important association for organism, because microorganisms can help the fitness of the host, because they respond very quickly, and so they can help to cope with the environmental changes. The Seagrass holobiont is still at infancy, I can say, and even more, the use of microbial communities as a marker of the ecological, ecophysiological status of the plants. We know some things, so that microbiomes are very diverse, that are different with different plant parts, and there is a quite good literature on single processes and mainly on sediment, but not so many are devoted to the study of diversity and composition of the communities. And so we have to do a lot of work, still a lot of work, because we have to identify the microbial patterns across different systems, healthy or disturbed, tropical versus temperate, etc., and how the microbial changes are linked to environmental condition, to host and plant ecophysiology. And last but not last, it's necessary an optimization and harmonization of all the experimental procedures, from the sampling to the softer bioinformatic pipelines to evaluate the data. Just to introduce you to this kind of study, I want to explain a little the way in which microbial community analysis is done. We have very powerful tool in next generation sequencing, that is the sequencing of Amplicons found in the sample, and this can give information on the structure and composition of the microbial colonizer present in each sample. This job is made of three main steps. First is the DNA extraction. Second is the true NGS. And then there is a lot of job made by bioinformatic tools. And this is a very rapidly changing field. So we start with motor, and then we pass to chime. What to do with these bioinformatic tools? What is possible to do after cleaning, we can say, the sequences that arrive as a FASTA file. It's possible to assign each read to the sample, and each kind of read can be assigned to the taxonomic entity, the more similar taxonomic entity, and then it's possible to evaluate on the entire set of data all the possible differences and similarities in the samples. So, on this base, we made several studies. So the first case studies that I want to show you are the first approach that we made to this work. And just to start, it was 2013 and 14, so very many years ago. We start by evaluating Nativa Lophila stipulacea in the Gulf of Aqaba to evaluate the variability of associated communities. And so we study a first case study that is reported here in this picture, by evaluating these three sites that are different according to geomorphological features and human use, reduced by, from this site that is North Beach to South Beach. And a second case study has been to study in South Beach what happens with depth. And so we use a single site and for depths for 9, 18 and 28 meters depth. What we found has been that by evaluating the morphometrics of plants, we saw significant difference in leaf area, in photosynthetic pigments and total phenol content among stations. So there is a change in the ecophysiology of the plants, and on the same plants what we saw is that there are significant differences in the microbial community composition between plant compartments and stations. So there is a sort of mirroring of the difference that we found in morphometric and biochemical descriptor also in the microbial community. And this is true also for the case number 2, that is the case of the depth gradient. Also in this case, we found differences in leaf area, pigments, and total phenols among the station, mirrored by differences of the microbial community, both above ground and below ground. So also in this case, we saw these differences reflected by the microbial communities. So, to summarize the case studies number 1 and 2, we can say that both morphological and biochemical descriptors show differences among stations, and this demonstrates the high plasticity of Halophila. And again, that microbial communities are significantly different among stations, so indicate also in that such a long time ago, we saw the possibility to identify possible functional relationship between seagrass and their associated microbial communities. Then we passed to other 2 case studies. Using a different approach, we studied the invasive Halophila stipulacea in the Mediterranean, and we found 2 places in which Halophila was in the same place with the native species, that was Posidonia oceanica in Cyprus and Cimodocea nodosa in Crete. And this has been devoted to evaluate the role of host plant and environmental condition on the microbial communities and the entire set of descriptors. What we found is that in the case of syntopic Halophila and Posidonia monospecific patches in the same place, both plants were suffered for this condition, and they showed a very high content of total phenol. Actually, the very high is in Posidonia, that is one order of magnitude higher than in Halophila. And corresponding to this, there are completely different microbial communities, as you can see, that are driven by the biochemical descriptors. Posidonia oceanica are the small tree spot on the left and Halophila on the right, so they are completely different. And what we found also is that each plant harbored unique microbes that are only associated to each species. In the case of Posidonia, for example, it has been interesting to find Burkholdereales that are phenol-degrading bacteria, and they are uniquely associated to Posidonia. In the case of the syntopic and patches of Halophila and Cimodocea that we found in Crete, the condition is quite a little different, because there are separate patches that grow until they became convergent in a mixed patch, and this situation has been found in 2 places, at 7 and 14 depths, 2 different places. And what we found is that the plants respond differently to the mixed condition, that is a condition of competition. And what we saw is that plant density decrease in Cimodocea and the increase in Halophila, so not affected. And leaf dimension decrease in both species, but in Halophila stipulacea at 14 meters, in which was higher in Halophila. So this showed that Halophila stipulacea may outcompete Cimodocea nodosa in morphometrics. But what happens to the microbes? And again, while in mixed patches, at both depths, microbial diversity decrease in Cimodocea and increase in Alophila. And this is quite helpful for Alophila that can support a very big and diverse microbial community. So, to summarize these cases 3 and 4, we can say that microbes are clearly different in each species, and Posidonia show peculiar microbes that probably are connected to the stressful condition, but highlighting the possibility of using these microbes as an indicator of stressful condition of one plant. Then seagrass synthropy, although is a quite rare phenomenon, showed us that it's a stressing condition for both species. High phenol content are found in both, and nevertheless this condition is much more stressful for Cimodocea than to Alophila, and in this synthropy, microbial communities are significantly different among seagrass, identifying a sort of higher capability of Alophila to harbor different kinds of microbes. So to conclude, we can say that epiphytic microbial communities thrives attached to seagrass and show differences between plant parts, so the environmental context shapes the seagrass holobiont. Actually, associated communities changes according to species and plant physiological status, and indicating that the host feature can shape the seagrass holobiont. The analysis of the microbial communities show that each seagrass under the same environmental condition can choose their microbes, so something like an elective affinity. And as last point, Alophila demonstrate a high morphophysiological plasticity and a high capability to harbor different microbes under different environmental conditions. So, this capability of Alophila stipulacea to host different microbes can be its ace in the hole to improve adaptiveness and invasiveness, and the superimposition of morphometric and biochemical results with the microbiome suggest the possible successful use of microbial community as a putative marker. And you can find our research on www.ecomicro.it, and thank you.